Good afternoon, everyone. Today's topic is explainable AI using SAP, Line, GINI, and Permutation Importance. So I would like to know how many people did explanation AI or doing a regular basis in your machine learning or data science practice. So anyone? Oh, you did. Okay, nice. Couple of people, right? Good. Okay. So before we start, we want to know that who we are. I'm assistant professor of data science at Florida Atlantic University. We are mainly doing the healthcare data science projects.
And I am, hello everyone, and I'm Subhosit Ray, and I am a postdoctoral fellow in data science at the Christine Lynn College of Nursing, Florida Atlantic University.
Okay, so this is the today's agenda. Before that, I could say that this is a 4 hours presentation before, and there are lots of coding we made for different other conferences like SciPy and CSUG and all those other conferences. But here we got only 25 minutes time, so I hope that I can complete this. We both can complete this. And there's a couple of things we can discuss here. Main important part: no coding at all and no videos. So in introduction to interpretability, that's the first topic, and then different interpretive methods. So we start with SAP, and then Subhosit will go Lime, permutation feature importance, and mean decrease impurity. That's called MDI. Let's go SAP first. So okay, little bit before that, I could mention who never use explainable machine learning. I could say that this is needed mainly for the black box model of the machine learning. So we do not know what happens, what kind of iteration happens there. And we generally get this kind of measurement matrices like accuracy, F1 score, and area under the curve. But we want to know why model is means model accuracy is maybe 90%, 85%. Why? What is the reason? Why model is success or not? So this is the reason we need to do the post hoc analysis. That is actually SAP, Lime, and MDI games. Post hoc is generally happens in a statistical analysis. So you see from here, there's two things. One is glass box where you know what you are doing. One is black box, you do not know what you means. You know that what you're doing, but that the stakeholder, they do not know that what happens means under the means in a model running. And these two terminology, interpretable model and explainable model, are interchangeable. A lot of people use this terminology, a lot of people use the other terminology. So don't confuse to see this. Those are means mostly same. So now another thing I could go before discussing the SAP analysis. One is a model agnostic, one is model specific. You see here, this is a model agnostic. The technology doesn't depend on which model you use. Like SAP, Lime, MDI doesn't depend on whether you use XGBoost or support vector machine or some other methods. But here, there's a model specific. So particular feature analysis technique or explainable AI, we can use particular for a model. Like for GINI importance, we use for ensemble model, like XGBoost or decision tree or random forest classifier. So let's go. So different interpretive methods. First, we come for the SAP analysis. So I'm going little details for why and how we use the SAP analysis. I will be little speedy. So if you want me, can tell me that I can maybe a little bit of get slower because a lot of slides are there. So this study you see here, the identifying patient characteristic for prediction of mortality of inpatient, the hospitalized patient with COVID-19 in South Florida. So this is the AI-driven decision support systems, and we use the black box model, and where we see 3 groups of data. One is patient characteristics, patient comorbidities, and medication, and we see the results. So we are data scientists. We can take care of what happens inside, but we have to connect to the stakeholder to make them understand that why model is reliable and you can believe the model. So our stakeholder is the clinicians. So let's move forward. So any data scientist start from understanding problem, then data pre-processing, data cleaning, data analysis, and results. So most of the time we use, and even the data is medical data is very messy data, so data cleaning and data analysis part. So this is the study we data was collected from Memorial Healthcare System in South Florida between March 2020 and January 2021, and those patients are hospitalized patients. And after testing RT-PCR, we 200 293 patients we excluded, and we got the 5,371 patients. And our criteria 10%, more than 10% missing value, we delete those columns. And now we have 25 variables, 24 independent variables, and one dependent variable, binary classification, survive, and mortality. And you know that other data scientists, we split the data 80-20 basis. So this is the data, so I'm not going details on the data. So how the data looks like, you see the two different color between the mortality and survived, and I'm just showing the three different kind of independent variables belongs to socio-demographic characteristics, three subgroups of patient age, four subgroup of BMI, and two subgroups of sex, and similarly pre-hospital comorbidities and medications here. So after that, we split the data 80-20, train and test. So you understand that what the problem here on the data sets. So data is completely imbalanced data sets. So this is the trained data sets, completely imbalanced data sets. What we did, we used the smart technology to balance the data sets, and we balance the trained data sets this way. So we increase the minority class and make the same amount of data on the with the majority class. But the test data sets as it is, because the real-time data set is always the imbalanced. And now, you know, after that, so random forest classifier, I'm not going details, so how the hyperparameter tuning, how it happens. So this is maybe the different kind of topics, but we use the random forest classifier, and this is the performance matrices. So until this, I believe that most of the people are familiar. So Mason correctly analyzed means 918 cases, and misclassified 157 cases, and our means AUC 76%, like a 26% more the stochastic level, like in a 50-50 chance. So Mason learned 26% times more than the stochastic level. Now this is the important case. So this is now the post hoc. So we use the two different analysis. One is SHAP analysis. This is the model-agnostic analysis, and random forest model specific. So random forest feature importance analysis, that's depends on GINI importance. So you see that the GINI index is written here, and the all features, 20 features here, so they didn't calculate the all 25 because other feature is so much less impacted. That's the reason they didn't do that. And features are in a y-axis, and they are in a ranking orders. So in both the cases, so the feature are in a ranking order, and this calculated by using GINI, and this calculated by using the Shapley values. And now you see that means how the GINI index is increased, and most important feature it is. So age is most important feature in this case. And if you see the other case, also age is the most important feature. Then BMI, then diarrhea, then here the race, and here the diarrhea. So all features are same, but similar, but not in the means same manners in a ranking order. That they change the place. So you might ask the why we did that, why we adopted both the technology. Because the precision medicine, we have to check that how the one another correlated. So that's one of the reasons. So here there's no information of the age. Somebody asked me that what age group is more important, what BMI is more important. So we do not know the answer from here. So we have to go this analysis called SHAP Beeswarms plot, and this is a dummy coding. So remember, this is not a one-hot encoding. If we use the one-hot encoding, we cannot correlate the data. It's a very difficult way. So I means encoded by hand. So how you read this graph? So this is the higher value. All means is a kind of confusion matrices. So all this cool color is a lower value and the red is higher values. And also, if you, sorry, heat map, this is called heat map. And this is, if you go here, so 0 is a neutral point, everything negative is supporting survival, everything positive supporting mortality. The binary class I already mentioned before. And after that, so if you see the age, so if you correlate from here, 0, 1, and 2, so 2 is a higher age group, so more than 65 years old, and that's denoted by the red color. And that's one of the reasons that who are the more, means, older age group people are more susceptible to die. So similarly, when you go here, is the BMI is overlapping of obesity and the overweight are more susceptible to die. But if you see that two medication group ACE and ARB, the kind of blood pressure reducing medicine is called lisinopril kind of medicine. You see the opposite direction. That means who are on the medication, they are less susceptible to die. And now that's a more important things comes here. Uh, so when there's a different plot, but SHAP analysis. So you see that first 15 features, how important that 15 features can give you 95% of model importance. So this is important because when the patient goes to the clinician might not need to check everything. So first 15 features gives you idea whether patient is susceptible to die or not. And these 15 features, if you see here, these 15 features, like first 15 features come from all groups with same patient characteristics. So 5 are from patient characteristic, 8 are from pre-hospital comorbidities, and 2 are from medication categories. So that means that that model learn from the from the data belongs to each category, and the way we can tell them that model is transparent and reliable. And now this is also important things that I mentioned that 76% time model is correct, 24% the model is not correct. So this is the individual cases. So before that is a global analysis. You see the global analysis, the total number of 5,371 patient. Based on that patient, I got this information. But it's a local analysis where we see the ID of the patient number, de-identified means cases, and two are two positive. One is two positive, one is two negative, and one is false positive, one is false negative. How to read this means data? If you how to read this means graph? You see that two different colors. So this color is supporting mortality, and this color is supporting survived. So these are kind of tag aware. So if you see here very small 0.500 is the resultant value. So if the value goes beyond that, so more than 0.5008, so this patient means died. So these two patient value is more than 0.5008, and other two patient less than 0.5008. So these are resultant values. So these two patients survive. This is a Bayesian prediction. Remember that. That's the reason I showed here actual and predicted, actual, predicted, actual, predicted. Two are means two cases means actual and predicted is same, and two cases they are not same. Bayesian falsely identified these two cases. And now this is the little difference from the different study. We actually, it's a COVID-19 patient, same data sets, but here this case is ventilation and ICU and IMCU are the outcome variables. The other was the mortality. And why I'm showing that? So before that, you saw the impact of one variables. Here, impact of two variables, diarrhea and diabetes. How that impacted on the outcome variables. Here diarrhea, diabetes, here age and hypertension. So you see that values here is all are SHAP values. So SHAP value more when that impacted with that other variable. That combined with the other variables that impact more. And after that, this is I took different other study. So this is the global analysis of SHAP, but in that case, I want to know little specific that during the two different gender, that as men and women, what happens. And we see that the SAP plots, the bar plot, they plot it differently, two different way. One is male for male, one is for female. And after that, this for information. So SAPLI, this is the full name of SAPLI additive explanation, and that is taken from means the persons who actually did it on economics data is called Lloyd Shapley, and who later won the Nobel Prize on Economics 2012. Okay, thank you.
Hello, everyone. So that was a sneak peek into SHAP, one of the most popular interpretable technique. And now I'm going to talk about Lime, which is an alternative to SHAP, and you can do a comparison between different interpretabilities if you so like. So I'm just giving you the tools for you to play around and learn more. So Lime is called local interpretable model agnostic explanations. It is developed by Marco Ribeiro in his thesis. It has a large number of citations, and as the model suggests, it's local. It's not like a global model. So what does that mean? Is that if you have a machine learning model that is separating, creating a threshold to classify your data over a complex landscape, you're going to pick a particular point that you want to explain how the model is deciding. And then you are going to fit a simpler model in that local vicinity by using some linear regression model. And then from the weights of the linear regression, you can identify which features are important. So pretty much that's the main idea. So here the glass box is the simpler model that you're fitting in the local vicinity using the predicted labels of the original model that you want to explain. Okay, so over here, I think it's called pip install lime is the library that you need to use. And this is the code that you are going to import from lime.limetabular. You're going to import lime tabular explanation. And then you are going to provide what are your class names, like mortality versus no mortality, or in this case, diabetes versus no diabetes. And then you're going to give it all the features that you have from the columns, so from the training data. And then you will be providing, I think, a particular ID over here. So this is like instantiation of the explainer object. And over here, you are going to give particular data set where you want to know how the model is deciding for that particular instance. And once you fit, you can pretty much ask the explainer to plot what are the important features. And it's going to tell you because the age ranges in this direction. So I give the model this much of this particular feature, this much of weight, which is 0.08, and so on and so forth. So these are again ranked in terms of the most important features. So first is the glucose, and then provided by age, then some other features. Okay, so that's the Lime. So what are the differences between these two? So Lime has some interesting features. So it's mostly made for local. So it has lots of tools for you to choose your glass box model. Like you can choose linear regression or a decision tree to explain that important features for that locality. But SHAP has both of them, and SHAP comes with a lot of, like, Beeswarm was one of them, like Waterfall Plot, where you can identify 95% of the features that makes the model's decision. And then there are many other things that SHAP has, but Lime does not come with all those things. Uh, so Lime is mostly model agnostic. It does not care what machine learning model you're using, but SHAP has both. It has different kinds of explainers. So tree explainer and linear explainer are model specific, and permutation explainer and kernel explainer, they are complicated. So they are model agnostic. And then Lime is mostly suited for smaller explanation like local area, but SHAP can handle a global landscape of data set. Um, and then Lime could be unstable because of several algorithmic choices. I cannot go much in details in here, but you can chat about it later after the talk. And then, like I said, SHAP has a lot of plots and ways to look into interactions and individual effects to the model outcome. So there's another one called permutation feature importance. And this one is not local, it's only global. And over here we have the idea is that like how to estimate what's the most important feature in the model prediction. So what is the idea behind is that, as the name suggests, you permute. So you permute the column for which the feature you are interested in. So for example, I'm interested in how much height plays a role in some sort of healthcare outcome or something like that. So what I'm going to do, I'm going to shuffle the values of that column so that in that permutation, you lose the information. Preferably, you are going to lose the information. That's the assumption. You might end up getting better prediction. So for that, to account for that, what you do is that you actually shuffle it k times and then look at how much the model has performing better or worse. And you kind of average over all the different outcomes. And this is the actual model without shuffle. And you are looking at the prediction changes over all the shuffle, and you take the difference. And that difference tells you if the difference is large after permuting, that means that particular feature is more important. And you are going to import this permutation feature importance from scikit-learn.inspection. So scikit-learn has that, pretty much the tool for doing model permutation importance. So the last one is mean decrease in impurity. Over here, you are using some sort of a decision tree. So this is model specific. So mean decrease in impurity is used in XGBoost, random forest decision trees, models like that. And you are pretty much sorting two different classes. For example, over here, I chose cat, species A and species B, but it could be mortality versus non-mortality or whatever your use case is. And you are going to sort based on some threshold of a feature for an independent feature, for example, like the features of the face or the dimensions of the ear size, things like that of the cat, and you're going to sort it. The idea is that how do I sort? The idea is that you are going to sort such that you end up getting more homogeneous in each buckets after the sort before compared to the root node. So we are going to select a threshold for particular feature like weight or some other BMI criteria. We're going to sort based on what threshold of that weight is a better sort of the dependent variable. And that's how we split the decision trees. And we keep splitting till we reach, sometimes it's not always like the first split you are going to get exact homogeneity. You are going to end up having still some impurity in each nodes. That's why you keep picking another independent variable and try to split based on a different criteria. So now if you have split based on ear length or things like that, then you can talk about facial width and things like that for identification of cat species or something like that. Now, pretty much you keep doing till you reach the leaf nodes, which are completely pure. So that's pretty much how random forest works. And the feature importance is derived from how much each independent features is contributing to the better homogeneity or better split. So if a feature is in the top and doing the most amount of segregation, then that's a more important feature compared to the rest. So that's pretty much the idea about behind decision nodes and the leaf nodes. And then the idea behind, now that's just one decision tree. Random forest is a collection of different decision trees. What you do is like you take an average of the important feature importance across all the nodes, and then you get the feature importance for a random forest. So that's pretty much the idea behind it. And over here, you are going to get forest. So random forest feature importance is embedded within any feature random forest classifier. So once you run feature importances like for random forest classifier from scikit-learn, you are automatically can pull feature importance out of the random forest, and you can plot it, and you can see what are the different important features, and their height suggests you the most important feature. So those are the three important things, and we have explored all these three. Mostly, we did not use Lime. We used other methods, the GINI and the MDI and the permutation importance and SHAP in this paper, where severity of disease, and we compared how it varies across different severities. And this is our mortality paper. So that's about it. And thank you for your attention. So if you want to go back to the QR code and scan, yep. And I'm happy to take any questions.